Okay, now we're on to carbohydrates. Name comes from how they worked out the formula originally. They noticed that there was a certain number of carbons, and then there was always twice as many hydrogens as oxygens. So carbo and then hydrate. Doesn't actually tell us much about its structure, but just sort of a historical thing. 3 types: monosaccharides, 1 sugar unit; disaccharides, 2 sugar units. Now these are the commonly occurring sugars, so glucose and fructose, found in lots of foods. Sucrose, common table sugar; lactose, milk sugar. And like I say, sugars, highly soluble, very sweet tasting. We can also produce polymers based on carbohydrates, and these are all based on the glucose molecule and many important molecules: starch, cellulose, glycogen being 3 of them. So first of all, monosaccharides, a single ring. Now, we look at the monosaccharides with 6 carbons in them. There are others we don't have to worry about. So when we draw sort of simple representations of structures, we'll draw 1 sugar unit, just as a hexagon, just as a simple representation of a monosaccharide unit. Now, we've got these ring shapes, and you'll see the way the ring is drawn, like this clearly is designed to be bigger, showing you the front of the molecule. That's the back of the molecule. We see hydrogens going down, hydroxyls going up in some positions and reversed in the other positions. These shapes are central to their function. So if it's OH down or OH up, that's very important. In fact, the difference between glucose and galactose, the only difference is at that carbon there, where instead of the OH going up in galactose, the OH goes down in glucose and produces 2 metabolically completely different chemicals. It can be a 5-member ring, still got 6 carbons, but 5-member ring, and that's fructose. And I think I've just answered that question. It's the arrangement around that carbon. Now, which carbon is that carbon? Well, we always number the carbons from the, effectively, if it's in the ring shape, from the end closest to the oxygen in the ring. So start here, go clockwise, 1, 2, 3, 4. So it's round carbon number 4. White crystalline solids. Because of all that hydrogen bonding from all those hydroxyl groups, it forms a regular crystal lattice. Glucose is so common in living organisms, the basic product of photosynthesis, basic energy source in cells, basic monomer for polysaccharides. Other monosaccharides are important, but not as common. Multiple chiral centres, don't need to worry too much about that. Highly soluble in water. All those hydroxyl groups can interact with water. So for a big molecule, molar mass of 180 with 6 carbons in it, they are really soluble considering that. Numbering the carbons clockwise from the oxygen, as I say. So 1, 2, 3, 4, 5, 6. Not sure you'll have to do that in the exam, but it does help you understand talk about the structures. Disaccharides, what we get when we join two monosaccharides together. Example, sucrose. So sucrose is a disaccharide made up of glucose and fructose. So it's a condensation reaction. We join together two hydroxyls, pull out the water molecule, join what's left over, and we end up with that carbon-oxygen-carbon bond, which we've come across a little bit in organic chemistry as the ether linkage. In biochemistry referred to as a glycosidic link. Join together the two hydroxyls, pull out the water molecule. Now note how if they are both in the down position, we are going to draw the ether link, the glycosidic link in that down position. We try and preserve that direction or that overall shape in our structures. Now, formal definition of a glycosidic link is where you join a sugar to an OH or an NH in another molecule for what it's worth. Other disaccharides, maltose. We've all heard of malt. It's the one that's probably the simplest, just two glucose joined together, but it doesn't actually occur all that often. Glucose and galactose, a more common disaccharide is lactose, milk sugar. So then the questions. Okay, two forms of glucose, alpha glucose and beta glucose. Now, because they're the ones that are close to the oxygen, the ring shape of monosaccharides alternates with an open chain form. So in aqueous environments, this happens all the time, interconversion between these two forms, the ring version, the straight chain form. So this straight chain form has got an aldehyde group on the end. Therefore they are known as aldose sugars. Sometimes the open chain form will have the carbon double bond oxygen as a ketone group. They are known as ketose sugars. So when they interconvert between these forms, when they form a ring again, the hydroxyl group on carbon number 1 can actually be in either direction, and so there's easy interconversion between those two forms, alpha glucose and beta glucose. Alpha glucose is when it's down, beta glucose is when it is up. Now, the three major polysaccharides are formed involving those different forms of glucose. So amylose is formed from alpha glucose, and starch is formed polysaccharides. Polysaccharides are polymers of glucose with large molar masses, so long chains of carbon atoms, molar masses in the thousands. Condensation polymer, water is produced each time a glycosidic link is formed. Now first of all we need to look at the two forms of glucose, alpha glucose and beta glucose. Now ring-shaped monosaccharides can actually interconvert into a straight chain, an open chain form of the sugar. And when glucose does, it forms this molecule with lots and lots of hydroxyl groups and an aldehyde group at the end. Now, when the ring joins up again, the hydroxyl group of carbon number 1 can either be down in the case of alpha glucose or up in the case of beta glucose. Now once you lock those together into a polysaccharide, then it becomes locked as either alpha or beta. So the three major polysaccharides, the first is starch, which is the energy storage molecule in plants. We get lots of our energy by eating plants with starch in it. Rice and wheat and the like. Alpha starch is formed from alpha glucose. Now there are two forms of it. Amylose, which is a straight chain form. So unbranched, therefore can coil up pretty tightly, make it harder for water to get in, which means it is less soluble. Amylopectin, on the other hand, is much more highly branched. That stops it from coiling up too much, meaning water can get into it, enzymes can get into it, it can dissolve and be broken down much more easily. So quick to digest, slow to digest. Glycogen is the energy storage molecule, the equivalent to starch in animals. Again, formed from alpha glucose, more highly branched than amylopectin even. Stored in the liver and muscles, hydrolyzed for quick energy. The third major polysaccharide is cellulose, the structural carbohydrate in plants, and because it gives plant cells and plants their structure, it needs to be much more difficult to break down. Formed from beta glucose, straight chains, closely packed with hydrogen bonds holding the chains together, giving it a strong and rigid structure. Very difficult to break down, requires special enzymes which not many organisms possess. So the summary of the different types of polysaccharide.